Welcome to all of you. My today's topic is leaf arrangement, morphology and structure. First, I'd like to introduce my topic. Typically, leaves are flat and thin organs of the plant and have one surface towards the axis, the adaxial or upper surface, and one facing away from the stem axis, the abaxial or lower surface. Most of the leaves are bifacial, having definite adaxial and abaxial faces, but sometimes they are unifacial, lacking such differentiation. These are commonly arranged on the plants in such a way so as to expose their surfaces to light as efficiently as possible without shading each other to promote the photosynthetic functions. The stalk of the leaf is called the petiole. The base of the petiole may have a narrow to broad point of attachment called the leaf base. A leaf that lacks a petiole is said to be sessile. In monocots, the leaf is always broadly sheathed at the base with edges either fused or overlapping. The internal organization of most kinds of leaves have evolved to maximize exposure of the photosynthetic organelles, the chloroplast to light, and to increase absorption of carbon dioxide. Most leaves have stomata, the opening and closing of which regulate the exchange of carbon dioxide, oxygen, and water vapor with the atmosphere. In contrast, however, some leaf forms are adapted to modulate the amount of light they absorb to avoid or to mitigate excess heat, ultraviolet damage, or desiccation, or to sacrifice efficient light absorption in favor of protection from herbivores' enemies. Among these forms, the leaves of many xerophytes are conspicuous. The shape and structure of leaves vary considerably from species to species, depending largely on their adaptation to climate and availability of light. Other factors such as grazing, available nutrients, and ecological competition may also influence the shape and structure of leaves. In most plants, leaves also are the primary organs responsible for transpiration and guttation. Leaves can also store food and water and also modified accordingly to meet these functions, for example, in the leaves of succulent plants and in bulb scales. Accordingly, leaves form the prominent food in the diet of many animals. This is true for humans as well. For whom leaf vegetables are commonly used as staples. Deciduous plants mostly occur in frigid or cold temperate regions, typically shed their leaves in autumn, whereas in areas with a severe dry season, some plants may shed their leaves until the dry season ends. In contrast, many other seasonal plants such as palms and conifers retain their leaves for long periods. Welwitschia retains its two main leaves throughout its lifetime, which may exceed up to 1,000 years. Not all the plants have true leaves. For example, in bryophytes, mosses and liverworts produces flattened leaf-like structures that are rich in chlorophyll. These organs differ morphologically from the leaves of vascular plants in a lacking vascular tissue, whereas the true vascularized leaves first evolved following the Devonian period, when carbon dioxide concentration in the atmosphere dropped significantly. This occurred independently in two separate lineages of vascular plants. The microphyls of lycophytes and the euphylls, that is true leaves of ferns, gymnosperms and angiosperms. Euphylls are also referred to as macrophylls or megaphylls. Dear students, now we will discuss the morphology of leaf. A structurally complete leaf of an angiosperm consists of a petiole, leaf stalk, a lamina, leaf blade, and stipules, small structures located to either side of the base of the petiole. Not every species produces leaves with all of these structural components. In certain species, paired stipules are not obvious or are absent altogether. A petiole may be absent, or the blade may or may not be laminar, flattened. The petiole mechanically links the leaf of the plant and provides the route for transfer of water and sugars to and from the leaf. The lamina is typically the location of the majority of photosynthesis. In monocots, the leaf is almost always broadly sheathed at the base with edges either fused or overlapping. In some taxa, such as grasses, poaceae, and gingeraceae, there is an abaxial flap or a ligule at the junction of sheath and blade. A leaf that lacks a petiole is said to be sessile. Two basic forms of leaves can be described considering the way the blade lamina is divided. A simple leaf that has an undivided blade and a compound leaf that has a fully subdivided blade. Each leaflet of the blade is separated and possesses the secondary vein. Because each leaflet can appear to be a simple leaf, it is important to recognize where the petiole occurs to identify a compound leaf. Compound leaves are characteristically found in some families of higher plants, such as Fabaceae. The middle vein of a compound leaf or a frond is called rages. Following are some important types of compound leaves. Palmately compound leaf. These have the leaflets radiating from the end of the petiole like fingers of a palm of a hand, for example, cannabis esculus, etc. These have the leaflets arranged along the main or midvein. These are of following types. Imparipinnate, odd pinnate leaves. These are with a single terminal leaflet, for example, fraxanus, rosa indica, etc. Number 2, peripinnate, even pinnate leaves. These lack these terminal leaflets, for example, Swetiana, cassia, etc. Bipinnate, which the leaflets are themselves pinnately compound, also called twice pinnate, as in Mimosa pudica. Tripinnate, in which the leaflets are themselves bipinnate, also called thrice pinnate. Decompound leaves, in which the leaflets are themselves compound, as in Docks. Trifoliate compound leaves. A pinnate leaf with just 3 leaflets, trifolium, laburnum, etc. Now leaf petiole. Petiolated leaves have a petiole leaf stem. Sessile leaves do not have a petiole and the blades are directly attached to the stem. In clasping or decurrent leaves, the blade partially or wholly surrounds the stem, often giving the impression that the shoot grows through the leaf. When this is the case, the leaves are called perfoliate, such as in Claytonia perfoliata. In peltate leaves, the petiole attaches to the blade inside from the blade margin. In some acacias, such as the kova tree, Acacia kova, the petioles are expanded or broadened and function like leaf blades. These are called phyllodes. There may or may not be normal pinnate leaves at the tip of the phylode. Now stipule. A stipule is present on the leaves of many dicotyledons. It is an appendage on each side at the base of the petiole resembling a small leaf. Petioles may be persistent, such as in roses and beans, or may shed as the leaf expands, leaving a scar on the twig and exstipulated leaf. Now venation. The pattern or arrangement of veins in the leaf blade is known as leaf venation. If there is only one prominent vein in a leaf, it is called midvein or primary vein. Branches from this vein are called secondary veins. Tertiary veins are produced from the secondary veins, forming a scalariform, ladder-like or reticulate or net-like pattern. Most frequent types of venations are: number 1, unicostate pinnate venation. Here the veins arise pinnately from a single midvein and subdivide into venilets. These in turn form a complicated network. This type of venation is typical of dicotyledons. Multicostate or palmate venation. Here more than one equally strong vein enters the leaf blade. It is of following two types. 3 main veins branch at the base of the lamina and run subsequently parallel, as in Plantago major. A similar pattern is especially conspicuous in Melastomataceae. Number 2, several main veins diverge from near the base where the petiole attaches and radiate towards the edges of the leaf. For example, acer, maples. Number next, parallel venation. Here the veins run parallel throughout the length of the leaf, that is from the base to the apex, typical in grasses. Dichotomous venation. There are no dominant bundles, but the veins forking regularly by pairs found in ginkgo and in some pteridophytes. Now we will discuss leaf arrangement. The arrangement of leaves on the stem is called phyllotaxy. The leaves may be arranged in one of the following patterns in a plant. Number 1, alternate. Here the leaves are borne singly and are usually arranged in spiral pattern along the stem. Alternate leaves are sometimes placed along the two sides of the stem, two-ranked or disticus, or on three sides of the stem, three-ranked or tristicus. Next is opposite. Here the two leaves are borne on opposite sides at each node of the stem. Opposite leaves may be spiraled as in red mangroves, rhizophora, two-ranked as in many zygophyllaceae plants, or decussate, the leaves of adjacent nodes rotated at 90 degree. The decussate condition is the most common arrangement among vascular plant species. Whorled. Here 3 or more leaves are attached at each node on the stem or appear to arise from the same level on a stem. Now radiolate. Radiolate leaves form the rosette-like structure. Now internal structure of the leaf, that is anatomy of the leaf. A leaf is a plant organ and is made up of a collection of tissue in a regular organization. The major tissue systems are the epidermis that covers the upper and lower surfaces. The mesophyll lies within the epidermal layer, is rich in chloroplasts, also called chlorenchyma. And number 3, venation, the vascular tissue, that is xylem and phloem. These three tissue systems typically form a regular organization at the cellular scale. First, we will discuss epidermis. The epidermis is the outer layer of the cells covering the leaf. It forms the boundary separating the plant's inner cells from the external world. The epidermis serves several functions. Number 1, protection against water loss by way of transpiration, regulation of gaseous exchange, secretion of metabolic compounds, and in some species absorption of water. Most leaves show dorsiventral anatomy, that is the upper adaxial and lower abaxial surfaces have somewhat different construction and may serve different functions. The epidermis is usually transparent, that is epidermal cells lack chloroplasts, and coated on the upper side with a waxy cuticle that prevents loss of water. The cuticle in some cases is thinner on the lower epidermis than on the upper epidermis and is generally thicker on the leaves from dry climates as compared with those from wet climates. The epidermal tissue includes several differentiated cell types such as epidermal cells, epidermal hair cells, trichomes, stomata complex, guard cells, and substrate cells. The epidermal cells are typically more elongated in the leaves of monocots and in those of dicots. The epidermis is studded with pores called stomata. Stomata complex consists of a pore surrounded on each side by chloroplast containing guard cells and 2 to 4 subsidiary cells that lack chloroplasts. Opening and closing of stomata complex regulates the exchange of gases and water vapor between the outside air and the interior of the leaf, and plays an important role in photosynthesis without letting the leaf dry out. In a typical leaf, the stomata are more numerous on the abaxial lower epidermis than on adaxial upper epidermis, and more numerous in plants from cooler climates. Now, mesophyll. Most of the interior of the leaf between the upper and lower layers of epidermis is parenchymatous, that is, it is also known as ground tissue, or chlorenchyma tissue called the mesophyll. Mesophyll is a Greek word that means in the middle of the leaf. This assembled tissue is the primary location of photosynthesis in the plants. In ferns and most flowering plants, the mesophyll is divided into two layers: an upper palisade layer of tightly packed vertically elongated cells, 1 to 2 cells thick, directly beneath the adaxial epidermis, Its cells contain many more chloroplasts than the spongy layer. These long cylindrical cells are regularly arranged in 1 to 5 rows. Beneath the palisade layer is the spongy layer. The cells of the spongy layer are more rounded and not so tightly packed, containing large intersource spaces among them. These cells contain fewer chloroplasts than those of palisade layer. The pores or stomata of the epidermis open into substomatal chambers, which are connected to the air spaces between the spongy cells. These two layers of the mesophyll are absent in many aquatic and marshy plants. Instead, a homogeneous thin-walled cells separated by large gas-filled spaces called arenchyma is present, which is meant for the gaseous exchange. Leaves are normally green in color, which comes from chlorophyll found in plastids in the chlorenchyma cells. In some plants, variously colored leaves are present and their color is determined by various types of chromatoplasts. Now, veins. Veins are the vascular tissue of the leaf and are located in the spongy layer of the mesophyll. The pattern of veins is called venation. The veins are made up of xylem, the vascular component that brings water and minerals from the roots to the leaf, and phloem. It is that vascular component which usually transports the sap with the dissolved sucrose produced by leaves in photosynthesis out of the leaf. The xylem typically lies on the adaxial side of the vascular bundle and the phloem typically lies on the abaxial side. Both are embedded in a dense parenchyma tissue called the pith or sheath, which usually includes some structural collenchyma tissue. I think all the parameters of the leaf morphology and its structure has been completed. With these words, I would like to take a leave. Thank you.